Very good morning to all. Uh, at the outset, I must thank my senior colleague and mentor, Dr. Sridhar sir, who gave this opportunity to come here physically and interact with you. I am not expert enough to deliver invited talk, but I am here to share with you whatever the knowledge during last 12-13 years I have gained in the field of climate change, insect ecology, and modeling in the context of climate change. So he has given me this platform to interact with you. So I must thank him, and I will try to deliver my best possible. Initially, we will have some basics of the modeling, why actually we should go for insect phenology modeling. I hope this is the second week of your training. Huh? Yes. This is second week of your training. So at least you are geared up with some basics. What exactly is the insect modeling? Why we need for insect modeling with respect to the climate change impact analysis. How many of you are already working with insect models or climate change and impact on insect pest? Any background anybody is having? Just to know the level of knowledge you have pre-training and then post-training, what is the advancement in your knowledge level after going through the lecture and hands-on. Anyway, the title is Advances in Insect Phenology Modeling, Rate Summation and Cohort Updating Approach to Understand Temperature Dependence of Insect Life Cycles. My presentation will be adhering to these points I have summed up, that what exactly is the climate change and how the thermal stress amplification. What is thermal stress amplification? It is nothing but global climate warming, temperature rise, what we are saying, and how it is affecting the agroecosystem functioning and abiotic and biotic factors and interactions in between them. Then ecological models for forecasting the pest activity. We have this one, so pointer. Then why phenology models? As my title says that we are studying the insect phenology models. Then why we should go for phenology models? What exactly is meant by phenology? Then how we can go for temperature inclusion in insect phenology? What is its importance? Then what exactly is the cohort updating and rate summation approach to deal with the insect life tables and simulation under climate change? So first we will have overview of climate change and thermal stress amplification. I hope you have been introduced with this. What exactly is the climate change? Can anybody tell me what is the climate change? What you understand by the term climate change? Because it is globally debated hot topic as of now for last 2.5 decades, if I am right. Okay, isn't it? So what exactly is the climate change? What do you understand by this? We will go two-way interaction. There should not be one-way flow of just bombarding the information from my side. We will have a session in interactive way. Don't get pressurized. I am not that much senior. I am not that much expert also. Whatever I know, I am sharing with you. At the beginning, I told. Sharing of knowledge, not delivering the expert talk. So climate change is nothing but change in climate of any particular locality, which is a gradual change and irreversible. Because whatever change has happened, that has happened. It cannot be reverted back. Hope you agree with me. And if it is happening at particular locality, but now globally everywhere climate is changing because of anthropogenic activities like afforestation, industrialization, increasing population, many other developmental activities. They are accelerating the rate of climate change, and that's why we are worrying for it. Because climate change is nothing new. Since beginning of this universe, climate is changing, but it was gradual and slow. But during past 100 years, and specifically during last 20 years, at beginning of 21st century, that is 2000 onwards, if you see there is dramatic or drastic change in the climatic situations, particularly rise in temperature, then floods, droughts, changing intensity of rainfalls, carbon dioxide concentration rise in level of CO2 in atmosphere and other greenhouse gases like so. So this is the scenario that how the global projected rise in temperature will be according to the latest assessment report of IPCC, Intergovernmental Panel on Climate Change. What it says that there will be average of 4.4 degrees Celsius rise in temperature of Earth's surface by the end of 21st century. Okay, and range will be like this, 1.4 to 4.4, based on different scenarios. Shared socio-economic pathways are being considered. Socio-economic developmental aspects of each and every region or country are being considered, and accordingly, the projection models are estimating that 1.4 to 4.4 degrees Celsius rise will likely to be predicted by climate change. And another thing that I already told, first 2 decades of 21st century, that is from 2001 to 2020, change that mean and max temperature, that amplitude between mean and max temperature is very large difference between mean and max temperature. Diagonal temperature variation and changing rainfall patterns are there. You can go through quickly this slide. This is the Indian scenario of climate change. If you see that these are warmest years since last 100 years. If you say, as per the IMD, India Meteorological Department, and annual mean temperature during 2020 was 0.29 degree above the average of last 100 years, if you see. Then two particular or very peculiar phenomena with regard to Indian climate is that winter warming. Have you experienced this thing? Winter warming? As I told, increase the difference between mean and temperatures. Day and night temperatures are differing. Very large amplitude is there, and diurnal variation will be there in temperatures. We are experiencing that winters are almost finishing very soon. There is no chilling winters now anymore, if you see, or the chilling period is very short period of time. Then what is the implication for insects? Implications for insects with winter diapause, there is very great implication. What that implication could be? Can anybody tell me? What could be the implication for insects that undergo winter diapause? Huh? Yes, definitely. Either they won't undergo or the delay in onset of diapause. Because winter chilling or low temperature that was not supporting the development was forcing the insect to undergo diapause. But now winters are warming. Warming means they are getting favorable for insects to survive or continue their life cycle. That's why there will be delay in the onset of diapause. Then another thing is that changing rainfall pattern. We are experiencing for last 20 years also how the pattern of rainfall is changing frequently and drastically. Prolonged dry spells between two rainfall events. Though there is onset of monsoon at the beginning, again there is gap between next successive rainfall event, and worse droughts we have experienced. There is increased intensity of rainfall, means in small span of time there will be huge amount of rainfall. Though the average rainfall of any particular region will remain same, but the distribution is affected drastically or dramatically. And that's why decrease in number of rainy days, increase intensity of rainfall, this will have implication for seasonal dynamics of pest. You know very well how the rainfall is affecting. Now that's why we need to study the climate change impacts. It is not moving here. Yes. So what could be the key challenges for ecosystem? Our agro ecosystem is having bifold pressures, mounting pressures. One side there is declining productivity and maintaining sustainability over long period of time. At same time, we have mounting pressure from increasing population to feed upon. So to bridge the gap between these two under the challenge of impending climate change, that interactive biotic and abiotic stresses, which altogether constitute 80% yield losses. If we consider the losses 100%, means whatever losses they are there in agriculture commodity, if you consider it as 100%, out of that 80% are accounted together by abiotic and biotic, if you can see. So challenge is that adaptation to these stresses is the key challenge for 21st century. So these are major concerns for pest management, that phenomenal changes in crop pests, frequent seasonal outbreaks we have experienced, resurgence is there, minor pest becoming major, this is very common, emergence and re-emergence. These are two different things. New invasive species are coming in our country as like Tuta absoluta, then fall armyworm and many more like Mealybug phenacoccus. And what is the re-emergence? Can anybody give an example of re-emergence of the pest? What is emergence and re-emergence? Re-emergence is that was already there in past, but it has again come down. Example is the pink bollworm in cotton, which was suppressed due to introduction of BT cotton. For last 15 years, it was not there, but for last 4 5 years, again, if you see, it is causing havoc in cotton growing belts, both in BT and non-BT cotton. Okay, so that is called re-emergence kind of things. Alarming signals for crop pest management, that aggravation of already complex pest problems, new problems we are already facing, and accelerated yield loss associated with the climate change. So these are some of the backgrounds or challenges. This is out of, because this is very big one. I will not waste my time because you know you want hands-on in insect life cycle modeling. So other lectures might have covered also, or they will cover in due course of time. Then what are the different models that we are using for insect life cycle studies? There are basically we are concentrated on dynamic continuous simulation model. That will be the population dynamic model. We are studying insect life cycle modeling, but these are different types. That mainly they are quantitative. We are concerned. Empirical regression. They will fixed relationship. They won't describe the underlying phenomena, mechanism underlying. Only there is one factor and another factor. Whether there is any change with respect to the dependent factor, that only describe. But phenomena governing the relationship won't be described. That's why simulation models or mechanistic models, where the mechanism governing the population change of insect with respect to the environment is considered. And as population is growing, that's why it is dynamic. So our models are dynamic one. And agroecosystems, if we consider the model, whatever models, crop simulation models, pest model, soil water interaction model, they all are dynamic continuous systems and dynamic continuous mechanistic models we are using in our system. So there are two broad, now I am coming down just from climate change to Indian scenario to insect pest impact, then different models, and again to the for insect pest activity. So these are two patterns for what we are studying the insect pest dynamics. I think yesterday or day before yesterday, you were told about Maxent, that climate pattern matching function. Has he told? Dr. Gundappa, one of my colleague, he explained about Maxent model. So that is come under category first, climate pattern matching functions. Okay, okay. But the principle on which the model is operating is the climate pattern matching functions. Means you need not have to detailed investigation data about the pest. Just you should have to have presence absence data. That is occurrence points of the pest from different localities. That is latitude, longitude data and corresponding climate data like maximum mean temperature, rainfall, seasonality changes, and other. Those are called as bioclimatic factors. 19 parameters are there, they are being used there, and you do not have to go into detailed insect life cycle study. But that's why they are just giving you about the potential distribution risk, not into the details of dynamics, pest dynamics, how it is affecting. That's why the second is most comprehensive, that is phenology models, or which are called as process-based climate response models, because species response to the climate is being studied here, and processes involved in describing the response are considered or taken into account. That's why phenology models are very important, and globally they are most accepted one. So we are concentrating on this part only. Now I have scaled down to the actual topic of our discussion. So the climate pattern matching functions already told to you, so I will not tell you that Maxent, Climax, whatever those models, they don't need any detailed data, but minimal inputs are the point occurrence. Wherever the pest is occurring, just get the report. Even from literature also, you can collect the reports that this is the place pest is occurring, and get the lat long coordinates and climate data from internet. That World Clim website is there. Data is available for global simulations. You can do it. So this is the procedure being adopted here. Other than Maxent and climate, there are GARP, then habitat, bioclim. These are different models. Those are called as climate pattern matching functions. As of now, Maxent most frequently why used? Because it is free of cost. Climax, Dr. Sridhar sir is the only expert, I think, in Climax, but it is paid. So that's why many people are not getting access to it as Climax. Otherwise, it is also a wonderful tool. So these are some of the examples. My own studies only with Phenacoccus solenopsis. how the climate pattern matching functions are being used to predict the potential geographic distribution, not more than that. Potentially, what are the suitabilities or different geographic areas for establishment of that pest? Okay. So this is again another study with the Tuta also we did. Now coming to the actual one. Now I will take more time here because that was just overview to I cannot start directly from here unless and until you are geared up with some background that phenology models or process-based models or climate response models. First of all, we should know what is phenology. Phenology is common to all, not only insect. Any living organism, phenology is there. What is phenology? What is phenology? Already given there. It is a critical timings of events in a species life cycle. If we consider our insects, then what are those critical timings? I have mentioned there. Moth emergence from previous seasons overwintering population. Very critical to us or not? From ecology of insect understanding, whether that emergence is important or not. And it is altogether governed by climatic factors because dipause initiation, dipause continuation, and dipause termination. Altogether, physiology of that particular insect and that to be modified by weather or climate factors. Then mating, after emergence, there will be mating between male females, how long it will go on, or whether it is single, that is unigamous or polygamous like that. Then oviposition is another critical time or critical event in the species life cycle to continue the further generations. Then egg hatching, very important, particularly in case of the insect, those are internal feeders, like the case of our pink bollworm or stem borers in case of other crops. Once larva enters the stem or ball, there is no use of spraying of chemical insecticides because efficacy will go down by almost 60 to 80% due to increase in accessibility of sprayed chemicals to the target site, isn't it? That's why these are critical events that we should target. And dipause, again, I am telling migration from one place to another, as in case of your fall armyworm or other migratory pests. So these phenology models, they are computational tools, just like your climax and others. These are computational tools that simulate the timings of biological events, means that simulate alternately this phenology of insect in response to climate. And that's why they are very much important to understand the response of insect to the climate change. And they use data on environmental variables to predict the timings of biological events for a particular species. Not only data, but detailed laboratory evaluations are required here, if you see. They consider the physiological processes and response of species. Physiological processes in the sense rate of development, survival, mortality in response to the temperature because of either activation or deactivation of the growth regulating enzymes inside the body. Because metabolic processes inside the organism's body are altogether altered by either rise or fall in temperature. That's why physiological processes we are saying it, including the effects of temperature on growth, development, survival, and reproduction. And they are used to understand the effect of climate on the timings of biological events, means the phenology of insect, and the potential consequences for agroecosystem and species interactions. That's why they are more useful to understand in more detail and to plan the management strategies. With climate pattern matching functions, detailed information cannot be taken, but they are handy tools in case of invasions, just like new species has entered in our country. So just to get the potential of that species, wherever it can spread, we can provide alerts based on to be ready with some new threat. But here, detailed investigations are required. And these are the some of the tools. Climax Dymax. Advanced version of Climax is Climax Dymax, where Dymax means dynamics of pest is included. That's why it is Dymax. And whatever the next one is that ILCYM, which we are having hands-on, insect life cycle modeling tool. The advantage with is that though it is phenology model, but it is free of cost. It is open source software, freely available and most widely used. That's why, because many institutes or many poor countries, they don't have access to the paid softwares. That's why ILCM as of now is most widely used. It is developed by International Potato Center at Lima, Peru, and most widely used. Then another is also there. Many tools I have just highlighted you. Then what exactly is different in these models than those, that they are more sophisticated and more progressive? Why they are more sophisticated? Because they need detailed laboratory assessment of life table of insect under investigation. The insect for which you want to study the phenology, study the insect ecology or changing climate response, you need detailed laboratory investigation and again field data to correlate or field data to validate. That's why they are more progressive, more sophisticated. They simulate the variability in development time of individual, then survivorship in each cohort and reproductive potential within the population using stochastic statistical functions. Simulation modeling has stochastic mathematical equations to uh, real-time simulation of the pest population. And that's why they are important analytical tool to evaluate, to understand and predict the insect population dynamics in agroecosystems under wide range of environment and management practices. Use in phytosanitary risk assessment also they can be used, because if you want to prevent the entry of any invasive pest into the new area, these models can be the risk assessment tool also. We will see. Then why phenology models? We have already seen that what is exactly the phenology. It's the critical timings of life events in species life cycle. Then if you talk of insects, whether it is single insect or is a group of insect, it's a matter of population. It's not a single individual that annoys you. You know? And if population is there, then population is heterogeneous at every point of time. Heterogeneous in the sense, egg, larva, pupa, and adult, four distinct developmental stages are there. And except certain insects having only single life cycle, this unigeneration, single generations like mango stem borer or white grubs and all. Others are all are heterogeneous population, because there will be always overlapping generation at any particular of time point of time in time and space. If you look into the population of Spodoptera litura, whether it is possible to get only one stage or all mixed population, egg, larva, pupa, moths, pink bollworm, or in case of mealybugs, white flies, population is mixed one, overlapping generations. Because of that overlapping generations, the mixed populations or heterogeneous population is there, and that population is altogether how that physiology I already told, then behavior, that is escape mechanism if the adversities are there, and biology, that is critical dynamics that we have discussed, and that altogether govern the pest dynamics in space and time. Getting my point? And that's why we need phenology models to predict the direct relationship between insect development and prevailing weather. Insect phenology models are nothing but they are giving you direct relationship between the insect development, means survival, reproduction, timings of emergence, and any other thing, and whatever prevailing environment there. So that's why phenology models are key component in understanding insect ecology. Now, why we need to go for inclusion of temperature in insect phenology? Forget about the slides. Can anybody tell me what is the importance of temperature in insect phenology? First course of entomology at level. Insects are poikilothermics. Means they have very limited ability to regulate the thermal environment, isn't it? Their body temperature follows directly the environmental range. And that's why homeostasis is not there like other warm-blooded or higher animals. And that's why temperature is considered as more about 70%, uh, whatever population is governed by temperature only among all other abiotic factors. Temperature is the key or dominant environmental factor governing the pest population dynamics, right from development rate, mortality, survival, reproduction, migration, everything. And that's why we have to have temperature as one of the key predictor among the environmental factors in insect phenology. And the models developed for insect phenology studying are having temperature as a major dominant environmental factor. If you see the limited ability, then what could be the, then increase in temperature, it will accelerate the rates of development, reproduction, and survival. We know that higher the temperature, lesser will be the time required to complete the life event. Is it not? And that's why capacity to complete more number of generation in short span of time. This is one of the predicted risk of climate change, that there will be more number of generations completed by insect than the existing under future climate because of climate warming. More crop damage, of course, because more generations, more populations will be there, more abundance will be there. Then increased temperature in summer and winter have two different implications. That winter warming, in first slide itself we have studied. That delay in onset of diapause. There won't be any no diapause. We cannot say that there will not be a diapause, but there will be delay in the onset of diapause because chilling hours are not there to meet the requirement of diapause. And because of that, low winter mortality. Because of chilling winters, many species, those who are cold sensitive, populations will die. Maximum population will die and survivors will enter into diapause. But as they are warming, environments are warming, then there will be low winter mortality because conditions are getting warmer. That means favorable to their survival. And increase insect survival. Ultimately, you know that early infestations and more crop damage will be there. But in case of summer, the case will be different. Wherever temperatures are already high, within the favorable range, there will be accelerated rate of metabolic development. But beyond that favorable range, there will be again mortalities. There will be non-linear response that we will see in next while fitting the equations. Then faster nutrient depletion means metabolic activities will be more faster. Early termination of diapause, isn't it? Because diapause will be terminated early and early infestation of the crop and more damage will be there. So these are some of the assumptions globally the scientists or ecologists have considered, and that's why temperature has been considered as a main predictor of insect development. And this temperature dependency of insect growth and development is principally applied in process-based phenology modeling framework to describe the basic physiological aspects like development, survival, and reproduction. That's why how the temperature has been included and what was its importance. The basics are like here. Now then, basics now over. Now we are actual procedure what we need to do before going to operate the software. What is the time now? We should not get... huh? Okay, still another half an hour with us. So if we have to develop the temperature dependent phenology model using insect life cycle modeling software or any process based phenology model, if you have to develop, so then what are the steps? Now practical part came. Before operating software, we should have to have some data. That data you cannot generate in a half an hour or 2 hours. The data takes 6 months at least to generate in laboratory, detailed laboratory investigation. Also, you need to have some past 10 years space data also to validate. So how data requirement is important to the quality output of model that you come to know here. Temperature-based phenology model, it has two main parts. First one is constant temperature studies in laboratory. Okay, you have to choose the temperature ranges at regular intervals starting from low to high, considering the ecologically relevant range for species development and the species which is under investigation. And you have to, based on that data, estimate the temperature dependency. Temperature inclusion we have studied. Then we have to fit the models for development rate, fit the models for mortality, fit the models for fecundity and adult senescence. Adult senescence means what? Huh? Death, not exactly death. Huh? What exactly is the senescence? Huh? It is aging. Somebody told age, but it's not age, it's aging. Process of aging. Because other immature stages like egg, larva, and pupa, they develop from one stage to another. Egg to larva, larva into different instance, and again into pupa, pupa into adult. But adult won't develop to the next stage. There is no further development, isn't it? So that's why it is senescence. It is aging. For immature stages, we have development rate. Where is that pointer? I think pointer is not there, no? Hmm? Anyway, no problem. Why we have used development rate for immature stages? Because they develop into the next stage. And temperature dependent rate of development is applicable to them. But in case of adults, they won't develop to the next stage. They lay eggs. That is different phenomena. That is oviposition. But there is no development of adult to the next stage, like the other immature stages. That's why it is called senescence or aging. At the end, they will die. That will be the adult mortality. But we have used senescence. Why? Because the longevity, particularly, or the duration of adult survival, will be affected by temperatures, and that temperature dependent longevity, how it is affected, either increase or decrease. That's called senescence, process of aging. We are also at the process of aging since our birth. We are at particular age. We are advancing our life because we don't have different stages like egg, larva, and pupa. Though we call it as a childhood, then young, then old, but physiologically or phenologically, distinct different stages we don't have like egg, larva, and pupa. It's just aging from birth to death. It's not. That's why senescence is considered in case of adult. So after getting all this, you will have your model. Develop. Huh, huh. That depends on the temperature under which you are investigating. है ना? It high temperature will reduce the longevity. Low temperature will enhance or increase the number of days to death of adult. That's why how aging is occurring at temperature. Aging, huh. It will be either shorter or longer based on temperature. That's the temperature we have to include. So once model is developed, again the question comes in mind. I don't know whether it came to your mind or not. Came to your mind that we have developed model at constant temperature, and this is not the case in case of field. Whether field conditions are constant, they are diurnally changing, fluctuating, dynamic, and that too under changing climate. If you construct the model at constant temperatures, it will help to estimate only the best temperature, then range of temperature, that is low and upper threshold, then optimum range, then also thermal constant requirements like degree days, then survival, mortality that can be calculated based on the constant temperatures. But it has to be validated against what? Fluctuating conditions to simulate the field-like situations. Then again in incubators or that plant growth chambers, you should have programmable incubators for this. That they should allow you to change the temperature at regular intervals, say for 15 minutes, half an hour, or at least hourly interval. Then those program you can feed in incubator and study again one set of life table in laboratory and revalidate these situations. And with this, again take actual field data. Actual field data I am taking, that field observations of occurrence of pest, then reaching at peak and declining like that. That validation has to be done with field level. That is very lengthy process, not to be but to the basics that the theme of modeling, how we can, and then how your model help in adaptation and mitigation to the climate change. It is already few of the things already explained to you in previous slides. Then what is, how many of you have already dealt, what is cohort? Age group means cohort is nothing but group of individuals of same age, है ना? Eggs, group of eggs is same age. All, if incubation period of egg is 4 to 5 days, egg less than 20 hours, you can consider one cohort, then three, है ना? At least there should not be significantly different durations. Then if larval period, entire larval period is 10 days, and we have 4-5 instars, then each instar can be considered as a cohort, isn't it? Cohort of individuals of same age means newly emerged larva and fully developed larva cannot be cohort. Okay. So just underline the individuals of same age is called as cohort. Then stages in insect life cycle, I have already told in previous slide that heterogeneous nature of insect population and age stage structured at time and space. Age stage structured means age will be also different. Stage will also be different. Some will be eggs, some will be larva, some will be pupa. And within that age of egg will be again varying. Age of larva will be again different. That's why continuous reproduction and overlapping generations will be there in insects with multiple generations, particularly. And to consider this, we have cohort updating approach. That cohort updating approach, what it does, it will track the development of each cohort independently and for mortality, development, and transfer of one stage to the next stage. How one stage is developing into the next stage, and what are the different phenomena like mortality, survivor, and development rate in that stage will be accounted independently for each cohort, each group of individuals, and that's why account for individual responses to the variable temperature conditions or environmental conditions. And that's why phrenology models based on this approach are considered more realistic. We are not consider generalized responses of the life stages. Cohort stages are being considered. And that's why if the 100 individuals of newly emerged larva, means neonate larva, we are taking 100 neonate larva, then response of each 100 individual will be identical to all populations of any insect. But if you take 100 larvae of different age and say that this is the response, that would not be logical. Because early instars are more sensitive, mid instars are somewhat immune, later instars are almost immune because they are entering into the pupal stage and like that. That's why cohort updating up. Getting my point? Now rate summation. My title says cohort updating and rate summation effect. What exactly is the rate summation? Already first sentence already over. That developmental response of insect to temperature is usually determined by exposing the insect cohorts to constant temperature, isn't it? Then usually insect development shows linear relationship with increasing temperature within the favorable range. Look at the graph at right side. Within the favorable means just see that dotted line which is linear response, and another curve is the non-linear model. Why we should switch to linear to non-linear, that is clearly seen here. If within favorable range, linear and non-linear models are almost similar, there is no difference. But where the problem comes at the extreme low and extreme high, where the linear model is not having flexibility to predict the variability. And that's why this deviation from linearity, it is called as rate summation. Deviation from linearity. We assume that it is linear, that with rising temperature, developmental rate will be faster. That is assumption. Earlier it was there before advent of the non-linear models. It was there that it is linearly increasing, but it is not the case. In case of plant physiology also, we have curves like that logarithmic phase, then stationary phase and declining phase like that, isn't it? For any biological activity, there cannot be linearity always. There should be non-linearity at extremities, both extremities. And that's why this deviation from this extremity, how to account for that deviation, and that is accounted by rate summation effect. So rate summation is nothing but, here we see, is called as rate summation effect. How it is accounted? So it is accounted like this. In order to reproduce the insect behavior identical to natural environment based on your constant temperature or laboratory temperature data, the development is approximated. At these extreme ends where it is deviating from non-linear, there are some mathematical equations or functions, that perpendiculars we are taking and the distance between the deviating lines we are calculating in mathematics. So likewise, similarly here, approximation is taken for this over discrete timing increments. Say for daily or hourly species, precise simulations. I told in incubator, you have to feed the temperature at different time intervals. And we have to simulate that deviation with this, and this proportion of development taking place at each time increment is summed till the insect is developed to the next stage. There is summation of development, means continuous development from 1 day to day 5 is not taken. Fraction proportion of development, if development is considered as 100, see that graph. From our table also you can see whether it is visible to you. The table is visible, not at all visible. This is your temperature. 15, 15, 20, 20, 27, 30. That constant temperature is not enough for this. Okay. Now, these are the days required for egg to hatch. So, low temperature, number of days are more. As optimum temperature, days are like this. And as you can see, this, days are like this. Okay. Now, logarithm you have taken. Just logarithm of this days. Then sample size is your 100. That I told you. Then development of sample. Then on this development is summed from here it is 0, then cumulative is taken 17, then 17 plus 267 is 44, 267 plus 333 is 77, and 0.33 100 is this is in percentage multiplied by. So from day 1 to day end, when the development starts and development ends for that particular temperature, if you see 15 is ending here, 100% development is completed. Development is starting here, development is ending here, 100% is completed. So proportion of each development at that period of time is summed till it reaches the next stage, means it hatches from here. Egg is hatching at this point. For 20 degrees Celsius also, egg development starts from here and 100% reach here. So this is the duration. So development is sum till the stage is entered to the next. And that's why it is called as rate summation effect at different temperatures. So this statement mean of your experiment individual yes. Individuals. No. 100 individuals, 100 eggs or 100 larvae at 15 degrees Celsius, they took 11 to 16 days. Means out of 100, some individuals hatch on 11th day, some on 12th, some on 13th day, and at 16, their entire hatching is complete. All 100 will not hatch on same day. No? It is range. It started from 11, it ended with 16. Is it not? Means this is the range, and you can take the mean also with this. And this is the logarithm is there. Then how the development out of 100, how many are developed on 11th day? 8. Sorry, on 12th day 8 are developed into the larva. Then next day 12, next day 15, next day 10, and there is 0. Means out of 100 only these many eggs have hatched, not all. And if you see 8 out of 100, 8 are hatched, and survivors is sorry, this is hatching 45, and dead is mortality is 55. Is it not? Sample mortality is 55%. Means out of 100, 45 has hatched at 15 degrees Celsius, 55 have died. Sample mortality, sample development. Getting my point? And the frequency of development, out of 45, 8 have developed. So 8 divided by 45 comes 0.17. Then 12 divided by 45 comes 0.26. 15 divided by 45 comes 0.33. 10 by 45, 0.22. So this is the portion of development at each particular day. And it is summed. This is cumulative frequency of 100. So 17 is there. Then this 2 will 44. Then again this 77. Again this up to here 100. So that is segmented development is summed till the hatching occurs. Now I hope it is clear. Any confusion? I understand it is very tough. I have studied everything for 10 years and I am expecting you to understand it half an hour. That is not the case. But just to give you the essence. And in future, if doubt comes, you can ask me anytime. I am there. But my purpose is to understand everything. So hopefully you have understood rate summation effect, how we are tracking the development at different time intervals and summed it till the entire cohort is developing into the next stage. Okay. This is the development as a function of temperature. Things. This is the calculation. This is data. Na? At 15 degrees Celsius, you have 100 individuals you have taken. Okay. So on 11th day, there is no hatching. But you found that on 12th day, 8 larvae has hatched. So you wrote 8. But just go one step back, 0. Because software requires that model data in that format. Okay. To differentiate between. Again, here 2 zeros, why? Separate between 20 and 50 marks. Start and end should be with 0. Just see here. 0. Start and end for 15. Again starting for 20 and end. Again 25 and end. Okay. So this is the data only. I will show you the data formats also while hands on. But how the calculations are being done at the background, software running is different thing. Many times we are happy that I could learn to drive the vehicle. Yeah. Just accelerator, pressing the accelerator, putting, changing the gear, and this driving the steering. We are happy. But what exactly when problem comes, vehicle didn't start, or in between some problem comes, then we are stuck up with the things. So running software is different thing. Just front end you are running, but you should know what is happening at the back end. You know, processes, and otherwise you won't be able to track the error, because software is a machine. It don't have any brain. Whatever data you enter, it will give some output. You will be happy, but you should know whether data was of quality and whether results are of accurate or precise. Otherwise it is just garbage in and garbage out. आटा पीसने वाली चक्की को आप चाहे तो चावल डालो, व्हीट डालो, ज्वार डालो, आटा ही देगा ना वो, देगा कि नहीं देगा? So it's like that only. Whatever you enter, it will give you some results. But don't be happy with that. First, error should come and you should be able to track the error. Then only modeling will be perfect. So shall I move ahead? So these are the basics of the modeling, not actually hands-on. Now come to here. This is the figure. Now you will get a clear idea about cohort updating and rate summation approach. Just see here, each cohort, that is egg, larva, pupa, and adult. Okay. Cohort you are tracking. Then from E0 to En, what is that? What is that E0? What is 1, 2, up to En? Huh? Yes. That in that chart I have shown you, na, 11 to 16. So this is day 11, for example, 0 up to 16 it takes. So daily, that is aging. Up to how many days it takes for eggs at that particular temperature to develop or to complete the development and enter into the larva. You know, that bold arrow again downside you are seeing. And what does this red side downside arrows? Daily mortality again. Isn't it? Daily, how many numbers are dying out of your sample, that will be tracked here. These red arrows. Yeah, cohort is there. You are updating the development and next stage. Again from here to again next stage. To next stage. And daily mortality. Okay. Now this period, that aging, this gives you rate of development to the next stage, because time required to enter to the next stage, based on that you will estimate the rate of development. So while doing these life table studies, whether at this particular, suppose take 15 degrees temperature, different stages from egg, larva, and all these things should be placed at that particular, or we have to study that, suppose egg we should go at 15 degrees Celsius from real first generation, and that larval stage it should be studied at next generation. I will show you actually the next step. Whatever question you have asked me, next step is that only. Just understand with picture or theme what exactly theoretically I have explained. Na? Rate summation and cohort updating. Just understand this with this figure. I will explain you. So getting it easy from here, whatever we have learned in previous two slides, rate summation and cohort updating. Cohort updating means one cohort is updated here to the larva. Then larval cohort is updated to pupa. Pupa is updated to adult, and adult again to the reproduction. Right? That is cohort updating. Means tracking the development of each cohort and updating to the next stage. And the summation means how aging and duration, proportion of development at that particular temperature from 0 to 100%. Till it completes the development and enters the next stage, that is rate summation. Very clear and very simple with this figure. But I explained you with the example also in previous slide. Otherwise, this is the entire theme of cohort updating and rate summation effect. Now, whatever you have asked, this is the slide here. This is the programmable incubator, not simple BOD or temperature. Here, besides temperature, you have relative humidity, then light regulation facility. They won't grow. This is the case with Spodoptera litera. I studied almost 6-7 years back. and different temperatures. These are the temperatures. At one time in one incubator, you can maintain only one temperature, constant temperature. Fluctuating temperature set is different. That is next step I will tell you. Then you have to track the development of each cohort, like from egg, larva, pupa, adult, then mating, oviposition, and again next generation. It is like that. So this is the procedure for data generation. Then how the data is to be collected like that. You just see here. For each cohort, you should have separate data sheet like this. This is for egg only. This is for egg. Then separate sheet for larva. If names are there, then if you want to go in instar wise, again separate sheets for each instar. Otherwise, you can keep one for that larva or name, no problem. Then pupa, female, male, and oviposition as a separate file because that file structure is different than this. Then column 1 indicates the batch or replication for same constant temperature. 2 indicate the temperature you are studying, that 10, 10, 15, 15 you are getting. Column 3 is the day when the event started, and another is the day when the event ended, and how many numbers, sorry, samples you have tested, that 100. So likewise, for each stage, you should have to have the data. Then from where to get the software? So this is, I told, freely available on the web. That is ilcym.cpotato.org. International Potato Center at Lima. They have developed. So here, first downside, green arrow shows that software can be manually in English can be downloaded from that horizontal green arrow. There is the manual. Okay. So this is the, now this is overview of the software. This is only front end view. As on you are went on processing and applying different processes, the views will change. But this is very front view. Immediately when you open the software, it looks like that. Project, modeling, compilation, simulation. These are different options available. Then your project, where it will be saved. Then what are the different variables you want to study, like rate of development, rate, time of development, mortality, like that. So this is the overview. Now, distribution of development time, I have told you that rate summation effect and all, how it is being calculated. There are different models. I think instead of theoretically going, now we will start practicals, because theory part almost I have covered. What are the basics of modeling? What is phenology? What are the rate summation approach? What is the cohort updating and all? Now this is actual calculations, how to perform. Instead of showing you simply the tables and figures, I think better to go with this. So I told you from where to download the software. If you have laptops with you, hmm, you want to operate on your own, or shall I give you demo first, understand it, and then practice it? I am comfortable with anything. I have no, huh? But I think demo will be useful because now you may simultaneously you may miss some steps, and again you will stuck up at that place now. Then it will be lag and it will be create mismatch. So theoretically, I won't go for this, because how we are estimating these functions practically, I will show you. Different. Already we have in theory we have seen mortality, development rate, development time, then fecundity, then age-related oviposition frequency, and different life tables. These are already known to you. That intrinsic rate of increase, finite rate, doubling time, net reproductive rate, generation time. These are common life table parameters, how they are estimated at different temperatures. So this is the um, picture graph and tabular this one. And now this one theoretical part was left, that how to validate the model. You should choose the actual weather station data from your locality, and for those season, this is the soybean pest I studied, Spodocara litura on soybean I studied. So during June to I think September or October, that kharif season temperature data, mean max data I have collected, and daily 15 half an hour temperature increment I have summed and put into the incubator. So the graph came like this, that daily at night 12 temperature will be like that. Then 11, 12, after that 1, 2, 3, 4, 5, like that. For 48 hours time steps we have given, half hourly temperature. And incubator was fluctuating the temperature as per the program that was given to it. And one life cycle set was studied at this temperature. This temperature range, DTF, is nothing but daily temperature fluctuation. That is the amplitude. That was representing June to October temperature, covering low, moderate, and high temperature range for insect development. We have chosen that one. And now this is the validation, means whatever data you have got based on your model, whether it is able to reproduce identically with that set under fluctuating condition or not. And that will be studied like this: predicted and observed values. They should be in agreement. There should be more than 70% agreement between predicted and observed values. Otherwise, model is not precise. And as it approaches to the 100, means 100%, it is more precise one. If the model with R square value 0.9 and above, they are more accurate. But in case of insects and actually field level data, if you are using, up to minimum 0.7% should be the R square value, or 70% should be the agreement between predicted and observed one. So this is the graphical presentation, that dots are your observed one and lines are your predicted one. So except that one dot, red dot in larva, that was outlier. Others, all our stages are more than 70% agreement was there in prediction and observed data. So likewise, you can tell you whether your model is working well or not. These are further recommendations also, what we can scale up, that we can use. This model has another beautiful feature is that it is coupled with GIS platform. So you can prepare maps of risk of establishment. This is another advantage with this process-based modeling. How it can be done, this is just briefly I will tell you, that we need climate data, both globally and for country level data is available. It's up to you whether you want to go for India level mapping or global mapping, or per point, means particular location, say for IHR location or any other location, you can per point simulation also can do and study the variability. Likewise, this is the index based on life table parameters we have estimated. Establishment risk means these are the areas, soybean growing areas, indicating the low to high risk of establishment. Green will be low, red will be high. Likewise, zoning, it is called as agroecological zoning, and this is important in case of phytosanitary risk assessment also. Okay, what is the risk of establishment, medium to low or high, likewise. Then I told that with rise in temperature, there will be higher development rate and shorter development durations, means more number of generations. That is more another prediction or hypothecation, that how it affect the, that's why we are able to estimate the number of generations the pest can produce in a particular region based on temperature conditions. And that can be represented in terms of GI, that is generation index. That is also possible. So establishment and generations will altogether give the abundance of population. That is called as activity. How the population will be abundant, low to high abundant or moderately abundant. Likewise, you can delineate. So this is advancement in the phenology. So these maps are how they are useful. It is given here that risk assessment will be can be done, and planning of management strategies can be done. Then per point simulations per location. For every location, you cannot map for individual location, because spatial mapping means entire data set we need. But for location wise also you can have. You are interested for as cotton I am doing, then north zone, south zone, and central zone we have considered. And representative locations we have predicted the potential growth parameters for the pest. Likewise, here in case of mealybug, that is in case of pink bollworm. And again, another advancement other than this, that coupled this with cotton phenology and predicted the number of generations at field level in case of pink bollworm, that was published in scientific reports last year. And we have validated it across India using this one. So how these models can be used as a tool in pest management, that quarantine measures as pest risk assessment already told, then developing effective strategies for IRM also, because number of generations and development of resistance has close relevance. It is described there. Then undertaking timely pest management actions like insecticidal applications, particularly in case of pink bollworm, before larva hatches from egg and enters inside the bowl. That is the timing. Critical timing you have to hit. Otherwise, your efforts will be futile. Likewise, these are there. Then further readings. In my lecture notes, I have given further readings. Though the notes are not very comprehensive, very brief I have given, but all further readings listed there. You can go through these kind of things if you want to go into the details. These are further readings with this aspect. So this was all about. But before that, I am really thankful to Dr. Sridhar sir and all other coordinators of this winter school that they considered me as one of the candidates to come here and interact and deliver the content on this topic. So this is great opportunity for me, and try to throw light on most of the important aspects of insect ecology and modeling in the perspective of climate change. Thanks to you all also for your patient listening for last 3 hours. I think. When we have started, I remember. So for 3 hours, we are continuously sitting without any complaints. In fact, it's a fruitful lecture, sir. I don't know whether complaints are still there, but they may be here also. We have not expressed it, but I enjoyed interacting with you. Thank you very much. You are most welcome for any other queries in future also.